Our final talk for the immunoengineering breakout session this morning is Professor Jeffrey Bluestone, and we're welcoming him back after his time on the faculty as a member and the former chair of the Ben May Institute of Cancer Research here from 1987 to 2000. In fact, when he left Chicago, it was such a big loss for us that the Chicago Tribune ran this article about how big of a loss it was and how maybe we might not even recover from this, right? But it's actually because of the leadership that he had in helping build such a strong department that we have such a multitude of collaborators in cancer biology, immunology, and everything. And formerly as a postdoc, I really read a lot of his group's papers, really groundbreaking work on how the immune system is able to kind of silence itself and basically regulate too much inflammation, and how this could be kind of misplaced in certain disease contexts. So currently, he's the founding director of the Immune Tolerance Network, which is the largest NIH-funded clinical immunology research program. And they're testing novel immunotherapies in cancer and autoimmune diseases. He joined the faculty at the UCSF as the A.W. Mary Margaret Clausen Distinguished Professor and is currently also the director of their Diabetes Center. As a member of the National Academy of Medicine and a fellow of the American Academy of Arts and Sciences, Professor Bluestone is world-renowned for his expertise in dissecting all of these basic processes that control T-cell activation and immune tolerance across all of these diseases. And recently, he led the Parker Institute of Cancer Immunotherapy as its president and CEO, and is currently also the president and CEO of Sonoma Biotherapeutics, which is a startup biotech developing cell therapies to cure autoimmune and inflammatory diseases. With that, I'd like to pass the mic back to you, Professor Bluestone.
Great. Well, let me just start by thanking the organizers for inviting me. But as was mentioned, I even though I live in San Francisco, I left my heart in Chicago. And anyone that says that Chicago hasn't thrived after I've left doesn't know much about your university, because of course you've done extraordinarily well, and especially the immunology and cancer immunology community, which I so much admire, has done an extraordinary job. So what I'm going to do is start my screen and hope that it works okay. Someone should speak up if it's not, and get my laser pointer going. So what I'm going to do today is to tell you about some of our work over the past decade, really trying to develop cell therapeutics as the next pillar of medicine to try to treat autoimmune and inflammatory diseases. Before I get started, just a few disclosures. As was mentioned, I am the CEO and president of Sonoma Biotherapeutics, now full-time, having retired from UCSF last December. And that company is very focused on regulatory T cells, which I'll be talking about today. I'm also on the board of Gilead, which owns Kite, which is a cell therapy company. And I'm on the board of Prevention, which is developing therapeutics to treat autoimmune diseases. So cell therapy, of course, has a centuries-long history, actually starting out in Chicago, where the first blood bank existed in Cook County, and really transformed health in humans by the ability to be able to replenish blood in people who have lost it. But most importantly, I think the transformation in the 50s when Donnell Thomas, then in New York, really developed the first successful bone marrow transplant, treating a twin with leukemia with bone marrow from that twin. And that really brought in the era of cells as therapeutics, with bone marrow transplantation really being a stalwart in many therapies for cancer. But I think it's fair to say that in about 2000, there really was a transformation in the way we think about their cell therapy. In particular, I think the sequencing of the human genome, which gave us an understanding of where the genes were and what the genes were that would control cell development, proliferation, survival, combined with the really growing ability to transfer DNA into cells, whether it be through viruses like lentivirus or adeno-associated virus, or more recently CRISPR-based electroporation, combined with this whole field of synthetic biology, which grew up out of the notion that one could actually engineer inside cells particular activities and attributes as you need it, has really changed the opportunities that exist for cell therapy. And with that has come really an understanding of what the value, potential value at least, of cell therapy is. First of all, it's a living drug. So in principle, you would give the drug once, and that drug will not just survive, but expand in patients. Cells are what we call polypharmaceuticals. They can perform multiple therapeutic tasks. They can deliver proteins. They can express modulating molecules on their cell surface. They can traffic to various sites. And it turns out that in spite of all the amazing human genetic variability, that when you develop a cell therapy, which most of them now are autologous, you can be pretty confident that in any individual, you can isolate the cell you want and put it back into that patient. And of course, the future is allogeneic and being able to use cells from one individual and treat 1,000 people. So I think that that is a very important aspect of cell therapy. The other opportunities, as I mentioned, are the ability to be very selective and engineered for multiple behaviors. We can deliver these cells either directly at sites or systemically. And as I mentioned, they persist for quite a long time, with evidence now in some of the early studies of T-cell therapies of these cells surviving a decade or more. So with that, in about 2013, there was an explosion in the field in cancer in developing T-cell therapies. And in that regard, there are now 4 approved drugs to treat leukemia and lymphomas. And we'll get back to a little bit more about what I mean by CAR T. But these cells are engineered to express specific receptors that recognize an antigen present on these cells, CD19. And with that, those approvals really has changed the landscape for cell therapy. And just to give you just some numbers here, on the left side is the number of clinical trials that are going on in the United States using either these artificially engineered receptors or the endogenous T-cell receptor that recognizes antigens. And you can see that, again, starting at about 2013, this has exploded with now hundreds of trials underway to test cell therapy. And the obvious implication of that is that if you look at cell therapy as part of what I would call this new generation of medicines, be they gene therapies, DNA, RNA therapies, or cell therapy, you can see this explosion in cell therapy that's happening right now. So all of that is to say that I think that many people believe that we really are on the cusp of developing a new type of medicine, that in addition to small molecules and biologics, we will now have the ability to use cells as therapeutics. And I would be remiss if I didn't point out that, of course, the University of Chicago and the Pritzker School of Molecular Engineering, especially with their new Chicago Immunoengineering Innovation Center, really has been at the forefront of many of the innovations that are now being adapted and moved into this new pillar of medicine. So I expect that Jeff and the other folks at Chicago will continue to be innovators and really change how we think about using cells as therapeutics. So what I want to spend my time today is really talking about one slice of that growing pie, and that's the ability to exploit cell therapies to treat autoimmune disease and organ transplantation. And I'm sure most of you appreciate that the human body is really constantly at war with itself. On the one hand, we're developing an immune system that's able to monitor infectious agents, be they virus or bacteria, to surveil cancers as they pop up, and to provide tissue repair mechanisms to help in settings in which tissue is being destroyed. So that's great. And the immune system has this extraordinary plasticity that allows it this ability to recognize anything. But unfortunately, the immune system can make mistakes. And those mistakes are recognizing self tissues when they shouldn't, when the immune system shouldn't be, or causing a uncontrolled inflammatory response most recently being evidenced in the pathogenicity of the COVID and the coronavirus and COVID-19, where really a lot of the morbidity associated with that disease is not caused by the virus itself, but caused by the immune system response to that virus. And so because the immune system has to live in this delicate balance of being able, on the one hand, to surveil and seek out and destroy foreign, recognized foreign substances, on the other hand, it has to be controlled to make sure that it doesn't cause a lot of damage within the body itself inadvertently. And so what the immune system has done is used many tools to try to avoid this overreactivity or self-reactivity and breaking what we call immune tolerance. But what was discovered really about 20 years ago now was that there was a small but very critical population of cells that are developed largely from the thymus, which is the T cell generating organ in the body, but also by other activities directly in tissues, leading to the expression of a key transcription factor called FOXP3. And that transcription factor, which when it binds to multiple DNA sites in the genome, will both be involved in the development, maintenance, and function of this very small population of CD4 positive T cells that control everything from autoimmune disease to allergy, has a role in organ graft survival, and then in a number of other settings in the body. And in fact, the experiment of nature, which led to this discovery by simultaneously, almost by Fred Ramsdell, Shimon Sakaguchi, and Sasha Rudensky, was the experiments of nature. And this is showing a young boy, less than a year old, who was born with a mutation in FOXP3, which, as in most patients, leads to a massive immune dysregulation, autoimmune disease, and death in most people, most boys. It's an X-linked chromosome by 1 to 2 years of age, unless the individual gets a bone marrow transplant to replace these Tregs that are missing in the baby. Similar studies done by Fred in mouse showed that a mutation of FOXP3 in the mouse, the animals all died within 3 or 4 weeks, again, from massive autoimmunity. So this put Tregs on the map as really a key controller of immune system biology. And as a follow-up to that, a number of folks, ourselves included, started looking at the state of Tregs, not in a genetic disease like IPEX or scurfy, where FOXP3 is mutated, but in general in other autoimmune settings. And what we found was that Tregs can be defective or deficient in a number of autoimmune diseases. And in some cases, it's a deficiency in the generation of the cells or survival proliferation of the cells. In some cases, they're unstable. They lose FOXP3 expression. And all of that leads to a imbalance in the immune system. But it's also important to note that in some cases, actually, the pathogenic cells that cause the autoimmune disease are becoming resistant to Tregs. And so that imbalance really has played a critical role in our understanding why autoimmune diseases in certain individuals occur. And a lot of this is genetic, although there's clearly also an environmental impact on this. And so let me just tell you why I think Tregs are, in fact, such an important master controller of immune tolerance. And it really is due to their polypharmaceutical activities, that they really do perform a number of different essential functions. They eat up critical growth factors that are essential for other cells to survive, like CD4 and CD8 effector T cells that are causing the pathogenesis and destruction. They make cytokines and other proteins that will suppress the local immune environment, like IL-10 and TGF-beta. They express on their cell surface receptors like CTLA4, which will actually shut down other T cells and effector cell and antigen-presenting cells from functioning. And they produce metabolic control factors that change the microenvironment that they're living in such that they become less inflammatory. And so when you put this all together, a Treg, when it's present in that local environment, will cause that environment to become tolerogenic or suppressed. And that's what we call bystander suppression, where one cell against one specificity can actually shut down a number of cells locally of multiple different specificities. And that bystander suppression, combined with this notion of infectious tolerance, that by changing this local milieu, Tregs actually recruit other cells into the site that become regulatory, leads to a very robust anti-inflammatory environment. And then the last thing I'll mention about Tregs, which is underappreciated, but really growing in its relative importance as this cell is being better defined, is the fact that this cell will also make factors that can actually participate in wound healing and tissue repair. Factors like amphoregulin, keratinocyte growth factor are secreted by these cells when they're activated, so that they actually will shut down, not just shut down inflammation, but also help to repair the tissue that was damaged. So obvious implications of this is that if we could harness the power of these Tregs, we might have a therapeutic opportunity in patients with these autoimmune diseases. And over the last 15 years or so, there have been hundreds of demonstrations of the power of Treg cell therapy in a variety of autoimmune diseases, in the mouse models like multiple sclerosis, type 1 diabetes, inflammatory bowel disease, and lupus. You can take these cells and put them into an animal and really ameliorate or even reverse disease in these mice. Similarly, Tregs have been shown to have a powerful activity in graft versus host disease, in organ transplantation, in this case, islet transplantation, where the presence of Tregs can actually prevent rejection of the islets, but even has had a role in inflammation in general. So in things like stroke or even ALS, adoptive transfer of Tregs into those mice that have those pathologies actually significantly reduces the disease progression and morbidity. And so when you put this all together, it really suggests that if we can harness the power of Tregs, we might be able to use them therapeutically. And so several years ago now, and several is about 13, I put together a group at the University of California, San Francisco, to see if we could build a clinical cell therapy program, a program that would test the ability of Tregs to actually alter the progression of disease, both autoimmune and organ transplantation. And we started, and as I go through the talk, I'll talk about all of these to some extent. We started with the idea that we would just take Tregs out of patients that have defective Tregs, which I mentioned earlier on, and see whether we could come up with a way to both isolate them and expand them so that we could get a lot more cells, hopefully functioning better, and put them back into the patient. And I'll show you some of our clinical data on this. This population, of course, gives us a very high cell yield, although the specificity is not selected. It's whatever the normal specificity of the cells are that we take out. And so its potency, it can be active, but it's certainly not going to be maximally active if we don't have a specific receptor. At UCSF, we have also developed approaches that can enrich for antigen-specific Tregs by stimulating them against cells that express an antigen. And we've been using this in the organ transplant setting. The cell yields are lower, but obviously the specificity and potency is potentially higher. And then finally, I'll spend the last part of the talk talking about this new space that we at UCSF, but as importantly, the Sonoma Biotech, thus the disclosure I made, is using, which is to engineer these cells to make them both more specific and potent, but also give us high yield of cells out of patients. And so this program started with an observation we made that we could find markers on the surface of these cells that would allow us to rapidly purify these cells so that we could get a population to manipulate. And what we found is when we isolated these cells and expanded them in vitro, we could get anywhere from a 300 to 500-fold expansion of these cells. But more importantly, at the end of culture, these cells looked a lot more like potent Tregs, not like the cells that we had taken out of the patient that were, as I mentioned, defective. They expressed higher levels of the IL-2 receptor, which is where the growth factor IL-2 works, the higher levels of this molecule CTLA-4, which I mentioned is a functional molecule on the surface of these cells, and ability to bind and modify TGF-beta, another important clinical immunosuppressive pathway. As importantly, if not more importantly, the cells we grew out were bona fide Tregs. They didn't change in their function or biology, but they also maintained their epigenetic fingerprint, so that the cells that we grew up were in fact not just bona fide Tregs, but quite stable in expressing molecules like FOXP3, which was key to their functionality. And we showed that increased functionality in two different ways. One is when we looked at signaling of the Treg. Here's an example of Tregs from a patient with type 1 diabetes, where the Tregs do not signal through the IL-2 receptor as well as in healthy controls. But when we stimulate and grow these cells for 2 weeks, now the expanded cells signal through the IL-2 receptor just as well as the control cells. And in a variety of assays, here's one in vitro assay, the expanded cells would suppress the effector cells much better than the fresh Tregs. And here it's demonstrated by the ratio of the number of Tregs you had to add to effector cells to see suppression. And you could see here, even at 1:32, you got significant suppressive activity, which was not happening with the Tregs directly out of an individual. So with that protocol in hand and that ability to manufacture these cells at high numbers, typically getting 1 billion or more cells out of the 6 million we purify from a unit of peripheral blood, we started clinical trials. And here's just a couple of data points from those trials. First, showing that when we labeled these cells with deuterium, which would allow us to follow them, we found these cells rapidly got into the circulation, and we could see that by the enrichment of deuterium in cells in the blood. And then over time, although they diminished over the first 90 days, even a year out, we could see a significant percentage of the Tregs still in the circulation of these patients. Perhaps as, if not more importantly, when we re-isolated cells out of the peripheral blood, we found that all of the label remained within this Treg population as seen here, and we did not see any label in other cell populations, suggesting that these cells were quite stable over that year and were not losing their Treg identity. So we did a clinical trial first in type 1 diabetes, in which we showed that if you treat patients, and we treated up to 2.6 billion cells in some of these patients, that in a significant percentage of these patients at 2 years out, they were making as much insulin as they were at the beginning of the trial, suggesting that we had altered the progression of disease, since most patients will, by the end of 2 years, have destroyed any remaining insulin-producing beta cells and have no more insulin production. Here we were seeing insulin production in about half of the patients. And this was similar to a study published out of Poland using the same technology to isolate and expand the cells that we did, and found that in patients treated with the Treg population, they were making more insulin at the end here of 1 year than the control group was at that time. So taken together, it suggested that the transfer of Tregs was safe, most importantly, and obviously the focus of the study, but might ameliorate some of the damage that was being caused by the immune system. The challenge in type 1 diabetes, of course, is you can't really look at the site. You can only look in the peripheral blood at insulin. And so we were keen to try to test not just other indications, but indications where we could look in the site. And so we chose 2 indications. One is pemphigus vulgaris, which is a skin disease, and lupus, a subset of lupus patients, which have cutaneous lupus, which also have skin manifestations. And that allowed us to do biopsies, taking a sample of tissue at the beginning of the trial before we put in the Tregs, and then 12 weeks later after we did the adoptive transfer, and to look at what was happening at the site in these patients. And what we found is that if this is the profile we saw at baseline in these patients, which is there were Tregs there, they expressed some level of CTLA-4 and FOXP3, but it was not dramatic. But if we then looked again at 12 weeks, we saw both an increase in the key markers of functional Tregs here, CTLA-4 and FOXP3, but also the numbers of cells in the lesion went up quite significantly. So that suggested that the cells were getting to the site, and that at that site they were maintaining, if not even enhancing, the phenotypic functional activity there. But perhaps more importantly, when we looked at the non-Tregs at that site, so again, same biopsy, but now we're looking at effector T cells that presumably were playing a role in destroying the tissue, we found that after the transfer of the Tregs, there were significantly fewer cells in the tissue. Here, CD8 cells were way down, and the percentage of those cells that were making pathogenic cytokines, in this case gamma interferon, was significant. I'm getting a chat. I don't know if it matters. I'll ignore it for now, unless it's a problem. And then what we also found in the lupus patient was it was true not just in CD8s, but in CD4 cells as well, that the production of cytokines went down quite a bit. So this was not unique to the autoimmune setting. We also did a trial in organ transplant. Here, these were kidney transplant patients that had been monitored over time, and at 6 months found to have evidence of inflammation, suggesting that the kidney might be getting rejected. And so at 6 months, we took Tregs from those patients, grew them up, and put them back in. And what we found is, compared to baseline, resolution of the inflammation in these 3 patients that we treated, and in some cases saw that even out 6 months with another biopsy. We also found that if you looked at pathogenic markers in the urine of these kidney transplant patients, that those pathogenic markers went down significantly. So overall, this data was suggestive, combined with our other clinical trials here, listed here, that we could in fact have a viable program where we could isolate, expand, and treat patients in a variety of different autoimmune and organ transplant settings with a safe therapeutic that indeed could have significant immunological effects. Most recently, and actually we just initiated this week, a trial in COVID patients in which we're putting allogeneic Tregs, these are Tregs from a healthy individual, into patients in the ICU on ventilators, trying to shut down some of the inflammation there, with the idea that one might be able to avoid some of the death that's associated with late stage COVID-19, and hopefully we'll know more about that in a few months. I'm going to just quickly apologize. I want to see what this chat is in case it's saying something important. It says I only have 5 minutes left, which is a little bit odd because I was told it was 45 minutes. But anyway, I will keep plugging along here for a few more minutes. So what I want to spend the last part of the talk on is talking about where we're going with antigen-specific Treg therapies. And what we learned early on is that Tregs can in fact work even better if they were antigen specific. And without going into detail of these animal studies, the bottom line is if we had a Treg specific for a single antigen, it could totally reverse disease. And so this was also true in the organ transplant setting in the mouse. So what we've done is we've now engineered Tregs to be able to express a chimeric antigen receptor, which on the outside has a recognition domain that sees an antigen, and on the inside a set of signaling domains. And this is the kind of approach that's been used for CAR T cells in the cancer setting. And we've chosen RA as our target because the science tells us that Tregs are defective in this setting. And so we believe an adoptive transfer works in the mouse model. So we believe it's an opportunity. And we've chosen an antigen that's present in the joints of patients with rheumatoid arthritis, citrulline vimentin. And the idea is if we express the CAR on these Tregs, that they will recognize their antigen, get activated, and shut down inflammation. And so we have a CV-specific external domain with the internal signaling going on here. And we know that CV is important in the disease. I won't spend a lot of time on this, but in every setting that has been looked at, the presence of CV correlates with activity. And so we've now made these CAR Tregs. We've been able to put them into animal models and show that these cells can actually respond to synovial fluid from patients with rheumatoid arthritis, recognizing the antigen in the synovial fluid. And we've even shown that these cells are capable of specifically suppressing immune responses by polyclonal activated effector cells in an antigen-specific way, only suppressing in the presence of the right antigen, but not presence in the wrong antigen. So we've done similar things in organ transplant, here having a specific CAR for a HLA antigen and showing that it can actually block transplant rejection. So the future for this CAR Treg therapy is obvious, is to take these cells, generate these CAR Tregs, re-put them back into patients and see what happens. So I'm just going to end by saying that there's a lot more to be done with this, because we can engineer so many things in these cells. And one of the things that we're working on now is engineering to make these cells more responsive to IL-2, their critical growth factor, because we know that Tregs are defective in responding to IL-2. And if we can improve that, we can, which we've done in the mouse, we can get much better functionality of Tregs. And to do that, and I'll skip our clinical trial here where we try to use Tregs, but to do this, we're using synthetic biology approaches, working with Chris Garcia at Stanford to engineer an orthogonal IL-2 receptor and IL-2 so that we can selectively expand only cells that express this receptor and not have a general effect of IL-2 writ large in the body. And so I think what we can say now is we're really at the front of this field. There's lots to be done. We need to be able to make a universal Treg to be able to give to any patient. We've got to be able to engineer a lot of different activities into these cells so they can function. We have to think and are starting to use different types of receptors that will recognize antigens in different ways. And so I try to make quick conclusions here, but just to say that this is work done by a tremendous number of people in the lab, of course, the clinical group as well. And I'll stop there. Thank you.
All right, thank you, Professor Bluestone. So we do actually have time for a number of questions here. First, from online, we have a few questions from Adrian Nauman. The question is, does he routinely sequence the protein portion of novel antibodies?
Yeah, so yes, so all of the, we sequence the DNA that we are using to make all of our CARs to make sure that we have a bona fide receptor that is going to recognize the target. But ultimately, the functionality depends on the recognition of the antigen itself.
If you would expect the off-target toxicity of a CAR Treg to be much less than the off-target toxicity of a CAR T in cancer, and thus if this could be a really promising area because of that reason.
If I expect off-target toxicity, and the two potential toxicity of a Treg therapy is, first of all, if these cells are overly immunosuppressed, they might do a greater activity that might lead to infections or cancer. We think that that's reduced by having a specific receptor that localizes the Treg function to a particular site. Second potential toxicity is if these cells become unstable and start turning on effector functions. Since they see self-antigens, they can attack self-tissue themselves. We'll know that when we get to humans. So far, the animal data looks good, and our preliminary data, our polyclonal Treg study, suggests the cells are quite stable. But we're also engineering these cells to maximize their stability. You can imagine situations like overexpressing FOXP3 to make sure that they don't lose FOXP3 expression, as an example.
The broad
So the next
versus
question I see here
California
from
ruling on
Adrian
CRISPR.
is about how I see the CRISPR rulings affecting innovation. I'm going to be quite optimistic here. My experience, whether it goes back to the early monoclonal days or whatever, is that if a technology works and you can make drugs with the technology, we will figure out a way to be able to license and utilize those technologies. So I think that most of the field is moving aggressively to continue to use CRISPR, and we'll worry about the legal stuff later.
Questions from the audience?
Yeah, it's a great question. So the first question is whether we're thinking about targeting Tregs as a way of eliminating the toxic effects of immunotherapy. We actually have a paper that we've just submitted in an animal model, work being done by Arabella Young in my lab, showing that some of the toxicity, one example of the toxicity with anti-PD-1 is type 1 diabetes. About 1% of patients treated with anti-PD-1 develop type 1 diabetes. And in this paper, we show that you can take islet-specific Tregs and put them into the mice, and they still reject their tumor, absolutely fine, but they don't develop the type 1 diabetes anymore. So I think that those kinds of opportunities are going to exist. I must say that I don't think it's I think it's a great experimental question. I think from a therapeutic opportunity, these are expensive drugs, and I'm hoping that we'll find ways of modifying the toxic side effects of immunotherapy without having to use a several $100,000 drug to do that. But certainly, in principle, it can be done. I want to thank everybody again for an opportunity to speak with you today.